The action potential arrives at the synaptic knob.
Yeah, that's a big...
Calcium channels in the membrane open, allowing calcium to move into the cell. Increased calcium causes synaptic vesicles filled with acetylcholine to fuse with the membrane. Acetylcholine is released from synaptic knob by exocytosis into the synaptic cleft. Acetylcholine is picked up by receptors on motor end plate of skeletal muscle. The sodium-potassium channels open, allowing sodium ions to enter the muscle fiber. Breaks down the remaining acetylcholine in the synaptic cleft. The increase in ions inside the muscle cell initiate an action potential on the sarcolemma. An action potential travels down the sarcolemma into the T-tubules and stimulates the release of calcium from the terminal cisternae of the sarcoplasmic reticulum, where it has been sequestered by calsequestrin into the sarcoplasm of the muscle cell. Calcium floods over the thin filaments and binds to the troponin, pulling tropomyosin off the active site on the actin. Purple calcium floods over the thin filaments and binds to the gray troponin, pulling the tropomyosin, which is red, off the blue active site in the actin. High-energy myosin heads leap to bind to the exposed active sites on the thin filaments, forming a cross bridge. The myosin heads pivot to the low energy state, releasing ADP and moving the thin filament over the thick filament, called the power stroke. ATP binds to the myosin head, causing the head to break free from the active site on the actin. The myosin head becomes energized as ATP is converted to ADP. Calcium is pumped back into the SR and attached to calsequestrin, allowing tropomyosin to once again cover the active site, resulting in relaxation of the muscle. The myofilaments once again return to their relaxed state, resulting in the relaxation of the entire muscle. This is a depiction of a sarcolemma, which extends from Z-disc to Z-disc. A myofiber is made up of many myofibrils. This structure also includes mitochondria, which generates ATP, T-tubules, and is covered by the sarcolemma. The fascicle is made up of many myofibers.